So what I was going to say is I'm going to use easy language because I know some of you aren't cognitive neuroscientists. I find that difficult to understand, but there we are. There we are. There are all sorts of people in the world. They make it richer and make it better. We're going to, and while he's still working at getting the camera going, this is a book. Actually, it's an atlas. And it's in French, so it will make you work hard, but it has lots of pictures in it, some of which you will see today, and can be bought on Amazon, but, you know. There's possibly an English version coming. There will certainly be a Dutch version, an Italian version, and we're trying to get a Polish version, but it's more difficult with that. Here we go. So we're going to explore the human brain with neuroimaging, and we've got to start at the beginning, of course. One can never start at the end. And a Neolithic skull, 6,500 BC, which is 8,500 years ago. As you can see, there's this wonderful hole in the middle of the frontal bone. You can tell that's a skull, can't you? Because those of you who don't know, those are the eyes, and that's the nose, and this is the hole. Right? The question is this hole a medical hole or a religious hole? We don't know. We don't know, really. It would be interesting if it was a medical hole. It's probably more likely to be a religious hole to let out evil humors and, you know, someone who was... It's a sort of early Putin type thing, you know. And then we have to skip a few generations. And this is Vesalius, who was probably one of the first people to, at least in the modern era, we don't know what happened before, to dissect the human brain and to look inside it and to see how it was structured, and to think about the brain, which was, you know, quite remarkable. Up to then, people had thought it was a bit of a vestigial organ. Of course, given the British election, it's possible it is a vestigial organ in some people. But, you know, and now what we have is this. So this is the anatomy as Vesalius would have loved to have seen it, because it's the anatomy in life of a normal, actually of a superhuman male. I won't tell you who it is, but it's pretty obvious, I would have thought. And it shows, this is seen from the front, this is seen from the side, and that's seen from the top going down. And you can see all the cortex and then the deep structures of the brain and the brain stem as it comes out through the hole in the skull called the foramen magnum down to the spinal cord. And that can be stopped. The important thing to remember about this is that these are pictures, but that's not what it's all about. What it's about is a matrix for the mathematicians. I'm being nice to mathematicians today. It's a matrix of about 100,000 picture elements, so numbers in a computer, essentially. And you can do an awful lot more by analyzing those numbers than just by looking at it. So, you know, if I showed you a brain of someone with a tumor, you'd see a boom, a hole. And that's what radiologists are paid for. So you could all be radiologists, really. But if you were they were looking at that, they wouldn't be able to say whether I had normal cognition or whether I was dementing and retired. Okay, so it's very important that, because it's taking us into the field of analytics. Because today we're going to talk about big data, we're going to talk about my intelligence, artificial intelligence, which is much better than mine, and all sorts of things like that. And the levels of resolution that we now have with imaging, when I started 45 years ago, imaging was nowhere near that sort of resolution. There were sort of smudges. But now everyone is doing imaging. So you can go from images which are plain microscope, such as here, with the cortex. That's the surface of the cortex. This is underneath the cortex in the white matter. You can selectively color the cells and show where their axons go. You can show those cells in layers. You can show them as clusters of cells and try and analyze how the different axons interact with each other and talk to each other. You can look with high-resolution electron microscopes at these little dots. Do you see here? These are the little synapses where there are actual connections, chemical connections between the fibers that connect the cells. So that's sort of roughly the level. Actually, we're better now. We can even see calcium ions going through channels in the membranes of those synapses. Thank you very much. So next we have the fact that we can now also scan fetuses with magnetic resonance imaging in utero. So that's this sort of stuff. And then we can quite readily scan infants in sort of little cocoon things, rather like that one over there. Young children who love the video games, and if you tell them that Darth Vader is coming if they move their head, they'll sit there for an hour or two. And then children, adolescents, these people who are like you, mature adults, and then the retirees. And what you see is that the brain changes with time. That's very important, because it's one of the first indications that the brain is a plastically organized thing. And this reorganization is one of the things that have really come to the fore over the last 40 years, because it's made us understand that the brain is not a fixed organ. It's something that's constantly changing. So the word constant, which I just mentioned, has changed your brains. For some of you, it's changed it profoundly. For some of you, you couldn't care less. But something changes in your brain. Everything you do changes strengths of synapses and so on. It makes you learn, makes you forget. It gives you emotions and such like. So the brain is highly plastic. And then the second thing about the brain you must remember is that we're talking about 10 to the power 11 or 12 cells. And that's sort of ignoring other cells in the brain, nerve cells, other cells in the brain like the glia and so on, which are we're beginning to understand actually have an important role in how the brain functions. And between them, there are something like 10 to the 18, 17, 18 connections. Now, 10 to the 17, 18 is sort of, except for physicists who now make computers that can make 10 to the 17 or 18 calculations per second, to the rest of us, this is meaningless. There's so much there. So that's the first thing. The second thing is that with magnetic resonance imaging, we can also not only look at the structure of the brain as a whole, but we can start looking at connections of structures. So there are 3 different types of motor cells over this part of the brain there, and they send connections down to the spinal cord, and we can trace them, and we can see how precisely they go there. And then the cortex itself shows areas which are not specialized. People used to have, you've all got one of these nice brains with love here and nastiness here and inventiveness here painted onto them, phrenology skulls. Well, they're completely wrong. This looks like a phrenology skull, but isn't it? It's talking about areas which interact with each other. It's talking about cells and systems of cells which obtain complex functions and which can reorganize themselves after damage. And we can look at these cortico-cortical connections in very specific ways. This is a particularly interesting thing. For over here, you have things to do with the motor system. So you can see the motor system occupies a lot of the brain, which are also occupied by things which involve the motor system. So I'm speaking to you, that's the motor system. And so we need to keep our concepts clear and clean. Here are deep structures in the brain. I showed you they're down here. See? And different parts of the cortex send fibers which remain segregated in these deep parts of the brain. So where do all these things connect? Well, it's probably quite difficult to connect up there where there are large distances. But here you see at these interfaces, it's probably much easier to connect and to integrate information between different systems to give us the sort of things that we do when we're being extremely intelligent. The other thing is that psychologists, neuropsychologists and psychologists, particularly psychologists, because a neuropsychologist is someone who looks at the brain after it's been lesioned, after it's had damage to it. But a psychologist is interested in the normal brain, essentially. So this has been an extraordinary tool for psychologists, because before they could say what they wanted, and if they had charisma, people would believe them, you know. But now they have to go and actually look for the anatomical and functional correlates of what they are suggesting in the brain itself. I have a former student who's now a very famous a scientist in Paris at the École Normale Supérieure, who has been creating these sorts of maps of systems that are related to emotion, perception of action, theory of mind, of course, which is important for autism research and such like. So things are advancing at an enormous pace in just the basic science of the human brain. Now, caveat, at the mesoscopic level. So at the level of a millimeter cubed at best. So if we could look at calcium ions going through ion channels in neuronal membranes, this is many orders of magnitude larger. But you cannot understand the brain as a human, as an organ, as a functioning thing, unless you understand it at all the levels of organization. And that was the function of the Human Brain Project, which is to try and understand the rules that organize the brain at all levels, right up to the psychological level, which is one that is trying to understand really complex things like interactions between humans and such like. For medicine, on the other hand, magnetic resonance is developing yet other ways. So instead of making the images of different types which relate to, say, this is a big tumor, you see, it has a broken blood-brain barrier, so it allows that stuff they inject you when you have your MRI scan to flood into it. Usually it wouldn't. Here you see the tumor itself with some of its blood vessels. Here you see other components, water, increased metabolism, but not terribly. You can see the fibers going around it, so the surgeon knows where to cut and where not to cut, and where if he would cut, he knows he's not going to be able to take the tumor out completely, but if he cut there, he would also remove your capacity to understand or to speak. So these sorts of the functions of the brain, these things are all now being used medically, and also things which aren't images. This is a spectroscopy, magnetic resonance spectroscopy, which is looking at the concentration of various amino acids and other molecules in the center of this tumor and giving you signatures of different types of tumors. For example, malignant tumors or non-malignant tumors. So this is rapidly exploding, and this depends on really good physics. So every good investigative MRI center needs really good physicists because they write on computers. They call that programming. And they give it, I don't think I would listen to them on YouTube, but anyway, they program and they make the MRI machine do different things and be sensitive to different aspects of the physics and of the structure and of the function of the human brain. Okay. The other thing that's happening with MRI is when talking about the pictures which we've been looking at for 30, 40, 50 years, no, much less than that, since the early 1990s, late 1980s, we're now also discovering that by using certain of these programmed sequences to capture the data of what's going on in the brain, we can make them sensitive to different things. Iron content. See all the iron here in the basal ganglia? Myelin content and the relative relationships between the amount of myelin, which is that stuff which insulates the nerve cells. It's like this black stuff. It's an insulator, but it's on an axon, which is the bit of the neuron that connects to other neurons through the synapses. It can look at myelin content itself, at water content, and not look at it, but more than that, goes quantitative. So you can make absolute measurements. So this is real scientific work, because you can make a measurement in me in Krakow, and in you in Timbuktu, and in you somewhere else, and they are comparable. And so we can start looking at populations as well as diagnosing individuals. And all that is in a quantitative manner. So all that's very important. So that's the background. So that's what I would call about 1/10 of my talk. Oh, we've only done 25 minutes, so you've got another couple of hours to go. Here we go. These are things now which are available to clinician scientists. All right? So I'm a clinician scientist. I've done all my life's work has been in science, but I've also done some neurology on the side. So because that is where I find my questions, and then I use and develop these techniques to try to answer them. And for me, as someone who's been doing neurology for about 40 years, at the end of my clinical professional life, I feel quite depressed. I feel as though I'm banging my head against a brick wall, and I have been probably for about 20 or 30 years. You know, I've had my ups and downs, but in the end, I know nothing more about the neurodegenerations of old age that lead to the dementias. I know nothing more about the psychiatric diseases that make young people invalids for the rest of their lives or have them kill themselves, or others for that matter sometimes. So those are the two massive, great challenges that are facing us, and we really need to start getting serious about trying to find out the causes of these diseases. Because as you well know, it's easy to take some aspirin to bring the temperature down, but if you don't take your antibiotic and you're suffering from a bacterial infection, you will die. So what you need is not just to treat the symptoms, which we do in psychiatric disease, and not terribly well at that, because we give lots of side effects, but you also need to treat the causes. And you have to be very critical about this, because for many years, many of you will know, the pharmaceutical industry has been pouring vast amounts of money into a substance called amyloid in dementia. I'm going to now to explain to you why this is a terribly bad thing and why they're pulling out, why they've wasted $50 billion over the last 20 years. And it just needs a slightly different mindset to that which is based on talking to people, getting their history, being compassionate, and then trying to help. You know, there's nothing wrong with those things, but if that's all you can do, then you're a clinician. If you're a clinician scientist, you're going to say, I don't know why, and I'm going to find out. So on we go. The first thing is just to convince you that this observation and diagnosis by clinical observation is, you know, it's okay sometimes, but when it comes to those classes of brain diseases, it's pretty crap. This is a clinically defined type of dementia, frontotemporal disease of the behavioral variant. Doesn't matter what it's called. It is a syndrome because it depends on information taken from the patient. By the way, that's completely wrong. Most of them come to you so demented that it's the wife, the mother, the lover, whoever tells you about it. So already there's a bias introduced there. And just following them over 10 years, this guy, Andrew Kertész, a psychologist, finds that the syndrome changes spontaneously. So if you're a doctor seeing this and diagnosing this, and you're another doctor who doesn't know about this diagnosis, you'll make another diagnosis. Semantic dementia, progressive aphasia, corticobasal disease, motor neuron disease. And of the same people, these arrows are showing the same people, after another 4 or 5 years, well, there's another proportion that will have changed a third time with their syndrome. So syndromic diagnosis seems to be a bit stable. We shouldn't be surprised at this because we've already decided that the brain changes with time. So depending on cause, this is entirely plausible. What isn't plausible, what we've all accepted, is that these are diseases, and they're not. They're syndromes. So that's the first big error. We know they're syndromes, so this is pathology. This is the gold standard. This is taking the brain out after they're slicing it and looking for the characteristics of all these diseases, syndromes, and finding no correspondence. All right, so this is something that isn't spoken about terribly much because it leads us into a state of increased anxiety. Now, the other thing that is happening in the world of medicine and in the world of research in general, of course, is the fact that we are not only getting these techniques of non-invasive monitoring of life in boxes which otherwise you can't see through, the various types of scanning, but we're also getting the major influence of modern informatics. And modern informatics is a fascinating thing because it allows us to look at all sorts of data which we haven't necessarily produced ourselves, but which we could use to generate hypotheses. So the detail of this is entirely unimportant. So if you like it, if you think it's pleasing aesthetically, please keep looking at it. If not, concentrate hard on what I'm going to say now. This is the work of half a day by a doctoral student who went to his computer and went to the National Library of Science in Washington and put in the names of all the anatomical places in the brain. And he asked to find relationships in papers that used anatomical names of places in the brain with genetic information across all the papers that the National Library of Science contains. And that was a quick bit of calculation by the computer, and out came this. Half a day. So the first thing you can see is that there are nodes, there are relationships, and you could begin to start thinking about what they mean. Now, they don't mean terribly much here, but there are some groups, some large groups, psychiatric diseases that have relationships not only to genetics, but also to other diseases. So already, you're beginning to find relationships which were unknown in the past. So that's part of the power of informatics. So right, where are we now? The clinical approach alone is not sufficiently discriminative. So you can't prognose well and you can't find good curative diseases. And doing the genes, it doesn't replace descriptive medicine, but it gives us a step forward. Now, at the time of about 1995 to 2001, there was a great big program called the Human Genome Project. You've all heard of it, I presume. And there's one fact that came out of that genome project that cost a couple of billion, which is absolutely remarkable. Before that, people thought mutation, disease. Huntington's mutation, which was discovered in the late 80s, Huntington's disease. Perfect. So all we need to do is find the mutations, find the diseases. Doesn't work that way. Doesn't work that way at all. Even with Huntington's disease, the one mutation can present in 5 different ways, 5 different syndromes. And then they will change over time as the patient gets older and goes deeper and deeper into the dementia of his Huntington's disease. The other way around, it works like that as well. So there's a syndrome called spinocerebellar degeneration. It's a degenerative disease. It's not very common, but it's well described. There are, at the moment, 25 different mutations associated with that syndrome. So whether you look at it bottom up, if you look at it top down, it's not easy. It's a complex thing. And you just need to reflect a little bit on that and say, well, that's pretty obvious. But it's remarkable how obvious things are not obvious when we go on about it. So what we're going to talk about now is let's use informatics and those images of anatomy and see how far we can go. Let's begin to test whether what I'm saying is correct or not. And what we're going to look at is those picture elements. We're not going to look at the images. We're going to look at the maths. We're going to look at the reality. And we're going to see if patterns of picture element abnormalities, and that's where artificial intelligence comes in, because that's what some of the most important artificial intelligence is doing, is discriminating patterns in highly complex data, whether it can be of prognostic or diagnostic information. So this is a bit complicated, but just let me take you through it. We're using a bit of machine learning as one of the components of one of the types of artificial intelligence. It doesn't work, this thing. You just hold it there, and then you're 5 slides behind you, and you don't know what you're doing. So I'll put that away. And what you're trying to find is a function which distinguishes one group from another. So it's a classifier. It's not a straight line function. That would be too easy. It's a highly complex function. You can't even image it because it's in multiple dimensions. All right? And what you find is that if you just get the best clinical diagnosis of Alzheimer's disease, doctors are making an error of diagnosis of Alzheimer's disease as a cause of the syndrome dementia, so disease causing the dementia. There are hundreds of things that will cause dementia, 30% of the time in the best hospitals. This particular selection is slightly biased. It's they seem to be super diagnosticians. In fact, that's been repeated many times. The world over, 30% error. Now, here's the great CEO of the great pharmaceutical companies where he got a pill for dementia. First thing wrong, That's a syndrome, that's not a disease. Well, you got a pill for Alzheimer's disease. Well, you're going to have some very big groups, because in your group of patients you're going to treat, your active group, you're going to start off with an error variance of 30%. Okay, so it's a bit dodgy. So we took images, and this is some time ago now, and it's the only study like this that has been performed so far. I wrote to all my mates around the world, I said, please send me images, actually not images, it was the files, of people who you have seen and imaged with dementia syndrome before life, and who you've characterized, and who have then had a post-mortem examination, and they have either had only the changes of Alzheimer's disease, so validation by pathology of Alzheimer's disease change alone, or they're completely normal. Because what have we found? We found people with the changes of Alzheimer's disease who were normal. Now, you remember I told you how redundant the brain was. It compensates. So amongst us, and amongst us of my age and Wojtek's age, we may well already be started on that little slippery slope, my friend. That alcohol we exchanged yesterday and drank, that may be keeping us in the, everyone thinks that actually keeps you up. But we may be on the slippery slope. But come back to that nice industrialist, the CEO for the pharmaceutical company. Now what we're telling him is that in the normal controls, which he's got in his great big random controlled trial, he has a 20-30% error as well. Because in people over the age of 75, up to 20-30% already have changes of Alzheimer's disease. So it's just crap, right? It's just stupid doing it that way. Take those images that I asked for. We're up to this sort of diagnostic rate, sensitivity, and specificity. This is repeated. This has been so we know it's true. One scan, one clinical scan, can do everything that the best hospitals do at the moment and increase your... All right, enough with this self-laudatory stuff, but just let me show you how they refine these diagnoses now. So those of you in the field will know what the ADNI database is. That's the Alzheimer's Disease Neuroimaging Initiative. And it's been going for about 10 years, and it's based in California. A couple of mates of mine are responsible for it, and they've been collecting barn door clinical cases and barn door normals age matched. And they're up to about, I think they're up to about 1,000 now. And they study them, and they follow them, and they And so this is this group, the clinical AD group. So I got them to send me 20 clinical ADs and 20 clinical barn door normals. Right. And what did we find? Using our support vector machine, which is a simple mathematical function, 15 of the clinical AD are AD with the pathologically verified mechanism, but 5 are not. So they must have a dementia from another cause, unsuspected. And if you take the clinical normal controls, 3 of them have the AD changes. Only 17 are really normal. 5 minutes, you're crazy. I've only got through half my slides. No, you told me an hour. Wasn't it an hour? It wasn't an hour? Oh, well, we'll move on fast. So, refining the diagnosis in life, which we will have post-mortem, and we would have had post-mortem, is extremely important. Now, the real problem with making informatics and medical research come together is old people saying, "Oh, rubbish in, rubbish out." By that they mean, well, you've put in all sorts of data which is incomplete, which is from different laboratories, which is uncontrolled, blah, 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 blah. The real problem is that the best statisticians in the world, like Sir Bradford Hill, already in 1965 were saying that the real advantage of big data is you don't need to clean it up and you don't need to have all the data there, as long as you have it all. And by that, I mean millions of bits. Now, you've all heard of the Higgs boson, haven't you? Yes? Who's not heard of the Higgs boson? Raise your hand. See, you've all heard of Higgs boson. Do you know how they found it? They found it very simply. They got these things in CERN, for which we paid billions in our taxes, going at very high speeds. They hit a target, and then there were lots of particles went off, and they measured them, and they started looking at their TV screens, and it was going. After about 2 months, and after 3 months, that's how they found it. They squashed down the error variance because it was randomly distributed, and they magnified the signal because it was always in the same place and always additive. So that's essentially what this says. The implications are that you can generate your hypothesis from big data. So now that's the really problematic thing for most scientists, because it means that Nobel Prizes are prizes for luck. It means you happen to have turned up at the right time, in the right place, had a little idea which went on to do great things, but it's nothing to do with genius. It's what we call genius, but it's not the essence of genius. It means that we can get all the data that we have, and we have large amounts of data everywhere. I'll show you in a moment. You could generate a set of hypotheses with probabilities by saying to your algorithm, which examines these complex data, show us what are the most likely models that explain the data the best when looking at linear correlations, interactions of first order, second order, third order, fourth, as many orders as you like. And then as you get your list, you then test them with standard techniques, scientific techniques, in the same way as if the hypothesis came from the mind of an Einstein. So that is the answer to the old guys who say rubbish in, rubbish out. If they say rubbish in, rubbish out, ignore everything else they say. They don't know what they're talking about, essentially. Some young guys say that as well, but they just want to be promoted. So we have lots of neuroscience data from the very lowest resolution to the highest resolution to the very lowest resolution. We generate millions of amounts of papers. So we're now generating 150,000 papers in this field per annum. So any medic or any scientist who thinks they could have all that information in there is obviously lying. So this is another good reason why you need excellent informatics, and a very good reason why you shouldn't publish in Nature or Science. You should publish your data immediately raw on the web, and then you should publish your first draft of the paper in an open access journal online. You should ask the editor to ask these people who are knowledgeable in the field to review it online with their names, and he can add some more, and you have a little debate online, and then when it's accepted, it gets published, because the way you're going to find this information is by typing into your computer, give me the papers that are the most cited for this and this problem. That's the way it's going. Do you know what the return is for people like Elsevier and Macmillan and so on for every dollar invested? 20 cents. That's 20% per annum, no risk. Why? Because we're vain. And because the rectors of our universities don't know how to assess us when we're up for promotion. They say, you first or last author? What if there are 50 authors? And what if there are 10 people needed to define a new algorithm? They're always stuck in the middle. How are they going to get promotion? I mean, it's just bonkers the way we organize ourselves. I can say this because I'm retired. The other big thing about bringing informatics in with medical research is that the standard mentally generated hypothesis, mathematically processed, confronted with the relevant data, there's a big bias there. Parameterization, optimization, what means fixing the model because the results aren't right, so you change the parameters to get the right starting points. Giving you a correlation is out. And we've got to go towards a bottom-up simulation-type modeling practice. Now, when I say out, I mean relatively out. And when I say we've got to go to this, not exclusively, clearly, but we have to be aware that we need to produce knowledge and causes, mechanisms and causes. We can't get that from correlations directly. So we have to change our practice. Data, masses of it. Hospitals, every hospital used, 5 to 7% is used for follow-up of patients. The rest is used by lawyers in order to take people to court. Otherwise, completely blocked off. No one can get at them. They're not complete, they're not structured, they're not standardized, they're not clean, and they're protected for privacy, protected against corruption. So it couldn't possibly be helpful. Well, I've just told you how it can be helpful. Imagine, just imagine, that you could federate all the databases in all of the hospitals of the world. You're going to say, well, that is really stupid. And then I would say to you, Google, 15 years. And you would be stupid, because that's what they did. They federated all the computers in the world in order to use their algorithm, which is used for purposes of advertising. So we could perhaps do the same thing for the purposes of clinical diagnosis with the algorithms adapted appropriately. But there are questions of privacy. We've solved all of this. In the latest European directive for personal, this is all solved. But all we can do is group studies. But that's all right. That's all right. When it comes to doing diagnosis on the basis of knowledge of group studies, you get the patient and the doctor together again, and the doctor gets your results. He says, do you mind if I send that to the big database which has all the group results to find out how close you are to the group, where you are on the new diagnostic schema, so that I can perhaps find a drug which is specific for you, real personalized medicine. Not the sort of guff that gets talked about at the moment. And then there's all these databases. I mean, just imagine if we federated all the world's research databases in the brain. Of course, you would have to deal with researchers. That's my data. That's the data that's going to win me the Nobel Prize. Rubbish. It's not data that gets the Nobel Prize, it's the thought behind it. And it's the thought of how you analyze the data. So thankfully, that's being solved now as well by the big grant-giving people, NIH, MRC, Wellcome Trust, French CNRS, INSERM. They're all saying you're getting public money, put your data out raw on the web, public money. It's yours. Very good. And this one, they all say to me, you're stupid. Well, again, you just have to think about it a bit. So I went to see this big CEO. Not the same one, not the other one that I was talking about, another big CEO. I said, I want your data. We were both holding champagne glasses, so we drank to that, and he smirked. I said, no, only the one from the failed trials. And he said, but that's about 95% of our data. I said, yeah, that's right. And then only the ones which are placebo. I said, yeah, of course you can have that. So I got 400 the next day. Different files like that on different normal subjects who had been given placebo in a failed trial. Why should there be? There's no use at all. I said to him, it's absolutely, you can't monetize that. He said, yes, but if we do what you say, we will monetize that because you want that data. We've got it. Right, clever, huh? And they could give it to us very easily. We've sorted out all the problems. We've sorted out the ethical challenges. There are some real ethical challenges here. I mean, people talk about ethical challenges as though, God was going to come down and tell you off if you've done it wrong. But it's not that at all. Is it ethical to underuse information? It's not being used, and we're paying for it. To misuse community resources, my money, your money. To retard the acquisition of knowledge by research, is that ethical? There are others there, but because she won't let me talk, I'm going to have to stop. So this is it. This is the end now. This is big data. But you can see it's not really, but it's getting there. It's 6,000 different normal people who are aging, which we got from big national studies in France. And they included all sorts of data, including genetics, proteins, analyzed in all sorts of ways, supervised by people who had experience. That's us. Biologically, by which biological indices or high-dimensional learning, no a priori assumptions. The sort of high-dimensional feature learning that DeepMind has done for the chess players and the Go players, and then there's that game, I don't know the name of that game, where not only has the computer beaten the world champion in each case, but they've now got a computer which has learnt from absolutely nothing from the computer that beat the world champion and then beat the computer that beat the world champion every single time. You know, I mean, we need to be using these tools, for God's sake. And what do we get? We get fabulous new pictures like the one I showed at the beginning. That is very possibly Alzheimer's disease because it's the largest. All the pink ones are demented. It has ApoE4, which is a risk factor. It has amyloid. It has lots of other things associated. And there are lots of other signatures, disease signatures, which cause dementia. And you can see that there are lots of different types of normal. And you think, this is looking a bit crazy. But of course, some of them are young. Some of them are compensated, so they look normal, but they're not actually normal. It looks as though there may be confusion between these. But all this is very preliminary. This is not big data. This is 6,000. We need 1 million or 2 million. That's why we need to get into the databases. And then we can visualize them in different ways, look at the relationships of the different abnormalities. Where it says rule, that means a disease signature, a pattern of clinic, anatomy, physiology, genetics, and all the other things, which defines best and groups a certain number of the subjects. And the different results that define these, that define the disease, because it suggests mechanism, are shown in different colors, and their relationships can be seen to each other. And then the final proof of the pudding is that if we take clinical Alzheimer's disease, dementia, so let's be clear, dementia, we think it's Alzheimer's, and normal, we think it's abnormal, and we integrate data across these 6,000, we find 4 groups. We find Alzheimer's with the pathology of Alzheimer's, dementia, what we think is dementia, but without the pathology. We find normals without any pathology who are real normals, and then we find normals, or what we think are normals, but with the pathology on the scan of Alzheimer's disease. And then we compare what happens to them over time. They all start off normal, and those with Alzheimer's disease become fully demented much quicker and much more than those who develop a dementia, which is not the Alzheimer's disease type, and then those that never change. So I'm just showing the two for clarity. This difference, right up to here, is statistically more important than the ApoE4 statistical difference. So by bringing all these things together, we should get very much further. These are the people who contributed. There were lots and lots, and I thank you for your attention.